And so just go back to this image, okay? Oh, let's see. Oh, I actually have to scroll back to this image because I have it a little bit out of place. So now what we're saying is, oh, we didn't just have a brown furred parent and a white furred parent. We have these parents of some regular mice, and they're going to have a puppy. And what we do is we collect that early preoccupation embryo outside, and we add some special cells that we've manipulated in a dish to that embryo. Then we're going to produce a chimeric embryo in the first generation, and randomly will be partly brown furred mouse and partly that cell is going to make parts of his body. Then when we breed it in the next generation, some of this animals will have sperm and egg that just make brown furred mice, and we don't need those. But some of them will make mice that are 100% the cells from the dish. So that's how scientists make transgenic mice, is they need to manipulate the genes in a test tube, put them into cells in a dish to find the cell that has the gene inserted, and then they can convert that into a mouse. And once they have that mouse, they can breed it forever. It's a new strain of mice, okay? So that first part is, how do you make a special mouse? And the answer is you take advantage of that, okay? And so that's the first thing these scientists discovered that they could do, is they could put a lot of energy into manipulating these embryonic cells in a dish, and then they could convert them into a mouse strain. It would take 2 generations to do it. They would make a first generation of chimeric mice, they'd breed that, and they'd find a mouse that produces sperm or egg that is the transgenic mouse, and they'd have it. So that's how transgenic mice are created. There's a somewhat newer method that's starting to take over, but this is a really satisfying one because it combines a couple of things. It combines this first story and the second story. Oh, and I guess they won Nobel Prize then, like that. And all they did is make them. That's all they did. It's just a strategy for making them, like that. And but once you can make them, you can make a lot. So at Maine Health Institute of Research, where students are doing internships, they have a basement with a mouse colony out, and they have hundreds of these mice strains they've made. They're transgenic mice. They can make a knockout mice, right, like that. So they can make different kinds of mice. And so we just want to dwell on that because mice are mammals like us, and there's this way to manipulate them and make specialized mice. So I'm going to talk about it a little bit. So the first part is just what we already talked about. That's how you can convert a special cell into a mouse. So we got that. You can ask questions about that, but we got that. And then the second part is manipulating these cells in a dish, okay? And so, for example, in the course videos, at the start of the semester, I said, well, here's a mouse. It's a transgenic mouse, and it's got muscles that glow red. The way it was made is through this strategy. The scientists took the cells in a dish, and they added a new gene that, when expressed, would glow red, like that, okay? And they actually added 3 genes that glow different colors, okay? So we're going to talk about, yep?
When you were just saying that, with the glowing muscle cells, those wouldn't get passed down, would they?
They would.
They would?
Yes, yeah. So again, remember the original story about the brown furred parents and the white furred parents, and they make these chimeric offspring, a mixture of brown and white random. But if you pick one of those brown and white mixed animals and it grows up and you breed it, it's only going to make white furred sperm or brown furred sperm or white furred eggs and brown furred eggs. That first generation is the only generation where they have these combination parents. And after that, that effect is gone because they never mix the genes together. And so after that, it's pure. They breed purebred. And so we can take that special cell and take it through that process, and we can find mice that are 100% that. Okay? All right? And just here is just a side note to this. Okay? And it's a crazy side note. So before the scientists had embryonic stem cells, so good, healthy, normal stem cells, this was back in, say, the 1970s, maybe 1980s, they could grow cancer cells in a dish. And there's a very unusual, very, very rare kind of cancer. It is a reproductive organ cancer, but none that you've ever met anybody that had. So it could be a kind of testicular cancer or ovarian cancer, but not, people get those. But there's a very unusual kind that's called an embryonic carcinoma or a teratoma. So a very unusual kind of cancer. And scientists could grow those cancerous cells in a dish, and they could use those and manipulate those and put those with the normal mouse, like the brown-furred mouse pre-implantation embryo, and they could get mice after a second generation that were 100% cancerous cells, like that. So that's the old school way of doing it. And of course, you're like, I don't know if I like that. You have a mouse that's just made from a tumor, a cancerous tumor that you put in a dish, grow in a dish, manipulate in the dish, add a gene to it in the dish, and you have a cancer mouse, like that, okay? Right, very unusual kind of cancer, different than any other kind of cancer, because those mice would not get that cancer ever. That cancer that's caused by that very rare kind of embryonic carcinoma does not involve mutations in genes. What's happened in those cancers is the DNA methylation pattern has gotten out of whack. And it can actually be fixed by sending it through that process. So that's a really advanced side story, but Bryce reminded me of that because this idea of are those guys going to be permanent? Yeah, they're permanent. And we're back to embryonic stem cells. They're healthy, they're normal, they're permanent. Grow them, sell them. So that's going to come up in one of our examples. And maybe we'll just jump in. Well, let's go through these. So we're going to learn 3 examples of types of transgenic mice scientists will make. They use the same strategy. The first part, we know how to convert it into an animal. And the second part we haven't covered, but that's manipulating it in the cells in a dish and adding a gene. So we're going to add a gene in the dish and turn that cell into a normal mouse. That's permanent variety. So the first one is just knockout. So we've talked about that. We kind of have a handle on that. And what you want to do, and the example would be that this is a good example because we know that, in the 1990s, nematode scientists discovered genes that were required for programmed cell death. And so scientists studying other species are like, so we want to study programmed cell death in mice and humans. So what they did is they used the computer to do a blast search, take what was the nematode SED9 gene, right? We don't remember that name, this nematode caspase gene, and just do a blast search to look for a similar gene in a mouse. And they found it, really good match, and they're like, this is so similar. I suspect that that's a caspase in mice, and we want to study it. And so instead of going out in nature and looking for mutations, because that takes forever, we're just going to build a new defective version of this gene, and we're going to use it to replace the caspase gene. So the first example is where we're going to take a normal wild-type mice, and we're going to alter one of its genes. And the gene we're going to alter is this one we think is a caspase, and we want to prove it by destroying it. So in a test tube, we're going to build, with DNA, we're going to build a version of that gene, a new allele, that just deletes the coding region. That would be a knockout allele, right? Caspase is a protein, and we just delete the coding region. It's a null allele, a knockout. So we do that, and we put it into these cells in a dish, and we use it to replace the normal caspase gene. And then we turn that cell into a mouse. And we study it, and we're like, yep, it doesn't have any programmed cell death. A knockout of caspase in mice has no programmed cell death, like that. And that's been done a lot because nematodes and fruit flies and zebrafish have been the basis of discovering a lot of new genes. And you're like, we don't care about those organisms, we care about humans. So we got to knock out this gene in mice because they're mammals and they're closely related to us, and we'll really figure this out in mice. So that's been done a lot. That's why those scientists won a Nobel Prize. Just that. Great, effort like that, okay? And so that's our first example. And we'll draw it out when we look at the details of the second part, but that's our first example. Then the second example is called a knock-in. So we learned about knockout and knock-in, and that's just a cute, silly name, but it's the official name. And the example like that is going to be about mice, and it's going to be about mice that are made in Jackson Labs, which is up by Bar Harbor. So if you're hiking around in Acadia Park, every once in a while you see these kind of spooky modern science buildings because Jackson Lab is gigantic. And they've been studying just mice for a very long time. And they make lots of transgenic mice using this strategy, and then they sell them. Okay, because they're permanent. Okay, and so here's an example of a knock-in. So a knock-in is where you say, we're not going to knock out the mouse gene, we're going to replace it with a different version. Okay, so we're going to knock in a new version of the gene. It's not going to destroy it, it's going to change it. And here's what they want to do. We got to set it up for a minute. So this just, the setup is this story. So all of you know that if a company's making a new medicine, they got to test it before they use it in humans, right? Test it to show that it's safe. And they're going to test it in mice, So we'll give this drug to mice, see if it works in mice, and then we'll start trying it on people, right? So that's very, very common. And if you want to get your new drug approved by the FDA, you're going to have data. And it's going to have to be that mouse data to show, yep, it works great in mice, okay? Mice are closely related to us, so those drugs perform, in general, really similarly. But there's some differences between a mouse and a human, and so sometimes that new drug doesn't perform exactly the same way in a human as in a mouse as it's going to in a human. And that's worrisome. And one of the common differences that happens is that the drugs are really often broken down in the liver. So the liver is kind of a detoxifying organ, and it has enzymes to break down foreign molecules like drugs. And those enzymes have orthologs in humans and mice. They do the same thing, but there could be slight differences in them. And as a result, the mice might break the medicine down faster or slower than a human will. And that's going to affect, the drug's performance in a mouse, and it's going to be different in a human. And that's a concern. Just how quickly it gets, gets out of the system. Too quick or too slow, right? And so we start with that situation. And the Jackson Lab scientists say, we are going to take one human liver enzyme gene, and we're going to knock it in to replace the ortholog in the mouse. And they say we're going to humanize the mouse liver. We're going to make it perform more like a human. And they'll knock in one gene, and they'll take that strain, and they'll knock in another. And they make it more and perform more and more like a human liver would perform. Okay? And it's a permanent mouse strain, and they call them humanized mice. And that's if you told your friends that, they'd be like, geez, that's scary. I don't want to go to Acadia Park anymore because they've got humanized mice up there, like that, and what the heck, all right? But it's their livers are getting humanized, just one gene at a time. And they're orthologs, and they're not knockouts, they're normal. They're doing normal. It's just going to perform more like a human liver and more like a human body, like that, right? So now, today, the FDA will say to drug companies, oh, so you want us to approve your new drug? All right, well, what's your data? Did you test it in mice? Yes. And the FDA will say, well, did you test it in humanized mice? And if the drug company says no, they just come back and they say, you have to use humanized mice because they're our best model. And Jackson Labs sells those mice for a lot of money. And other businesses do that as well. Yep.
Do they humanize mice in other ways to
Yeah.
test for specific?
Yep. Yes. Yeah. And and because they can just start swapping in these genes. And again, it's not a knockout. Knock-in is a cute but a satisfying name. We're going to knock in a new gene like that. And they're just using the old knockout, concept, and they're saying, we're not knocking it out. We're not destroying it. We just want to make it perform differently, right? And one way is to just insert an ortholog from another species that would perform a little bit differently. Yep. Okay? So that's an example, a knock-in mouse. So we've got knockout and then a knock-in. And both of those involve replacing a gene. And then our third one is the muscles glowing. And that's a different story because in that story, we're not going to replace any genes. We're going to add a new gene. So that mouse from the video that's glowing, it's got all its normal genes. It's going to develop normally. It's not going to have a mutation at all. It's just got an extra gene. And that's it. So we could replace genes, or we can add a new extra gene. So those are our examples. And so here you see, and we're going to talk about this, both of these are created in the dish by having a genetic process occur called homologous recombination. And then this one involves non-homologous recombination. And we don't need to learn the details of that, but homologous means similar. And so these, we want to replace a gene. So we want to have that gene inserted and replace an existing gene, and it's homologous, it's similar. Right? So we want to destroy that caspase gene we're trying to put into that same spot. So we require that homologous recombination. Here, we're not trying to replace anything. It doesn't need to be homologous. It can be non-homologous. Just insert it anywhere. Get it into the chromosome so it's there permanently, but anywhere. Okay? So that's a significant difference that we just want to be square on, like that. Homologous recombination is going to replace an existing gene. Non-homologous is going to add a new gene. Okay? Like that. Okay? And so this was just, talking about the caspase ones to remind you of that. And so there, back to that first example, we have this gene, and then we haven't studied these, but there's a similar gene, so let's knock it out and see what is the effect, what is the phenotype when we knock out that gene. Okay? And here's just a picture, and it's from a book, I don't remember, in the third or fourth chapter, that's the old edition. And they're saying, here's how we create a knockout. So, and this is the part in the dish. We got these cells growing in the dish. They're embryonic stem cells. And we're going to design a gene in a test tube. The DNA is in the test tube. And then we want to get it into the cell. And here it's just showing we designed the gene so that these blue parts are similar, homologous, and we'll have homologous recombination and we'll replace the existing gene. So that's what's shown in that drawing is an example of homologous recombination occurring. Getting the DNA into the cells is very difficult and very rare, maybe 1 in a million times. And then getting it so the DNA goes in through homologous recombination is also rare. So that's why we're working in the dish, because in a dish we can have billions of cells. And we just have to get 1 out of a million, so we can get that done. It's very rare, it's not natural. And so that's why we're in a dish, is because we couldn't just put this into a mouse and say, go for it. It's just very, very rare. These are not natural steps like that. And so we're working in a dish with lots and lots of cells, and there's more to the story about how do we identify the cell, that rare cell. How do we find that? And we're not going to cover that in our course, but I'm happy to answer questions about it or talk about it separately. But how do we find that kind of needle in a haystack cell in a dish? How do we get that? And then that's our special cell. So again, the first part is how are we going to turn a special cell into a whole mouse permanently and freaking sell it, Sell it. Because you could, the company buys, the drug company buys it, is gonna pay a lot because they now have the special mouse and they never need to buy it again. What's the price tag? I don't know, I have no idea, but it's a ton of money, right? Developing a new drug is incredibly expensive, and this is one of the steps, right? So the first part is, how do we take a special cell and get it into a mouse? The second part is, how do we create the special cell? And that's just, for us, especially, well, do we want to do homologous recombination and replace a gene to make a knockout version or a knock-in version? And there are other examples of that, if you want to learn. Or we're putting in a new extra gene, glow or something. So homologous recombination versus non-homologous. So those are the two parts, and that's really what we're going to get out of that. And the book, is showing you this and talking about it, right? And it comes up in that early vertebrate development. And it's just this side story. And it's this like, oh God, I can't believe we're going to talk about this, right? It fits into that early development and the differences between vertebrates, right? They start from very different eggs. And mammals have no yolk, no maternal factors, have the early pre-implantation embryo. All the cells are equivalent. You could split them and make twins, or you could glue them together and make a hybrid chimera. So that's just an unusual feature of mammals. And we're mammals, right? Like that. And mice are where this is normally done. That's that, okay? And I think that's the total thing there. The book talks in different places in more details about it. Because again, these scientists, of course, are probably still all alive, and they're like, we're cool, we came up with this. And it's not just big business, it's big research. Like I said, down in Scarborough, they just have hundreds of these. They're just making them all the time because they can do so many different experiments on these various transgenic animals they make, okay? So that's the first one. And now we're just gonna go scroll back to the other two things I wanna cover because I got the slides a little bit out of order. So the first one was to talk about that, about making transgenic mice. And we have the two parts to that. Here's the next one. And the next one is actually covered in chapter 1, and we haven't talked about it yet. So it's a big, big deal, and it's just one word, and the word is morphogen. So that's going to be a fill in the blank, on our next exam, morphogen. And what it is morphogens are the sum of these signaling factors. So we have signaling factors like Wnt and sonic hedgehog and so forth. Some of those can function as a morphogen. And a morphogen is just a certain specialized version of a signaling factor, okay? So sometimes they can function as a morphogen, okay? And a morphogen is a signaling factor that causes different responses in target cells depending on the concentration. So that's literally what it is. So we've talked about signaling factors like Wnt, and we're like, and the cell's going to respond. What we're adding is, with a morphogen, the concentration of the signaling factor affects what the target cell does. And there are some incredibly famous examples of morphogens. That's why it's in the first chapter, okay? And so we're just going to draw that out. And the way that the book likes to draw it out, and developmental biologists draw it out, is they like to say, okay, here's a source cell. And it's going to make a signaling factor, and it's going to release it. So we're just going to make a graph. This is just a cartoon graph of the concentration of the signaling factor. And then this is the distance from the source cell. And the signaling factor is going to be at a high concentration near the source cell. It's going to decrease as you go away from it. So then there are going to be some cells, and these are going to be target cells. And you can see this target cell, this target cell, this target cell. And there could be more than that, but these are target cells. And you see some are closer. So this target cell gets exposed to that much of the signaling factor. This one gets exposed to that much. This gets exposed to that much. This is exposed to that much, okay? So different amounts. And if this is a morphogen, those target cells will make different responses. And easily the most famous example, I should have put one more cell in there. And there are more cells in this. This is a cartoon. But here's easily the most famous example, and that is our fingers fit into this story. And when we hold our hands, the pinky is the posterior and the thumb is anterior. And we'll learn late in our course that there's some cells in the posterior region of all vertebrate limbs that are making a morphogen, and the morphogen patterns the identity of the fingers and toes. So, each of our fingers and toes has a different identity, like pinky, ring finger, middle finger, index finger, thumb, okay? And that's based on seeing different concentration of this morphogen, and this morphogen is called the sonic hedgehog. And it's a signaling factor that can do this sometimes, where different target cells do different things depending on the concentration. And the source cell in this example are some cells in the limb, in the very early limbs. Like, our early limbs are just little tiny paddles, smaller than your fingernail by far. Really little tiny, little buds. And that's happening back then. And this source cell or population of cells, they're called the polarizing region. Because they set up the posterior to anterior axis, not just in our hands and feet, but all along the limb. All along the limb, there's an anterior and posterior side to it, and it's been polarized, it's been organized like that. And so this shows up in our first chapter on morphogen because you see the power, the potential power of signaling factors in what? In regulative development, in causing cells to become different from each other. And sonic hedgehog does that over and over again. It's one of these toolkit tools. It's a signaling factor. It does lots of signaling, and sometimes it functions as a morphogen. Okay? So that's this example. And it comes up again and again and again, like that. Okay? All right? So we're going to talk about one example of where sonic hedgehog plays a function like that as a morphogen in all vertebrates. And it's a cool example because at the same time, there are two other signaling factors functioning as morphogens. So the target cells see kind of a triangulation of signaling factors, and they make a lot of different cells. And the example are the somites in every vertebrate. And so remember the somite, in cross section, somites, we draw them like that. There's a somite, and it's bilateral, so there's a somite. There's the notochord, there's the lateral plate, these are like that, right? Okay? Okay? So these somites, and remember, they start out as like a long, solid finger, and then they get sliced into little segments. So we have these individual segments like this, where that's one somite, that's another somite, that's another somite. And each somite is going to do something different, like that. So that's a somite, and so the somite's shown there. And we're going to redraw this now and make it bigger, and just slide over here to show this effect. I don't know why we're showing that. I better just double check. Dang it. I think that didn't ruin our current recording. Just gotta check that. Yeah, we're fine. That was just a leftover from when things messed up. So I think we're doing fine. So we're gonna just redraw this kind of familiar drawing. We're just gonna look at one side. So we're gonna look at the notochord, the neural tube, and should make these different, but then I'm going to make the somite pretty big because we're going to focus on that. On the lateral plate mesoderm. And so this is the somite, and we're looking all along the body length at each one of the somites, and they're going to start to get patterned, right? They start out as just little chunks of a hot dog, and we're like, now we have to do different things. We're going to have to make some muscles and some bones. We're going to have to make some dermis that's under the skin, like that, okay? And the early step is just to start to break it up, and we're going to see that we're just going to talk about the somites becoming two different things. And one of the regions is normally called the dermomyotome, and it looks kind of like that. And it's actually composed of two parts. Part is going to make the dermis. The dermis is under the epidermis, so throughout our body, the epidermis is made by the ectoderm, and then the dermis underlying the ectoderm, epidermis is made by mesoderm. So there's the dermis, and then the myotome can make muscles. So the myotome is this region here. And then there's a third region, we'll say, that's called the sclerotome. And the sclerotome makes some cartilage. And it's going to contribute to bones also, but this is just a very early starting to get kind of a division of labor going in these somites.
What's the purple called?
The what?
What's the purple?
The purple is the myotome.
Okay.
Yeah. So muscle precursors. And although we just see these first names coming up, there's lots and lots of details. The cells become very different from each other, but we're just seeing a little, kind of broad determination. And what we're going to see is we're going to see that there is a signaling factor coming from the notochord and part of the neural tube, and it's going to make one of these, graphs like that. So concentration coming from these source cells, and the target cells are the somites. Then we're going to see another one coming from the lateral plate mesoderm, and doing that. And then we're going to see another one coming from another part of the neural tube, the upper part of the neural tube, and making like that. And I know that looks really busy, and that's fine, but it's not just one morphogen. We have three morphogens, and each target cell in a somite is responding to all three. So you would say like this, wow, the sclerotome is made because it sees pretty high concentration of this morphogen and very low concentration of that morphogen and low concentration of that morphogen. So see, we can add those up to make a really complex thing and get a lot of different cells. And these are all precursors long before they become muscles and bones and cartilage, where we start setting up their fate and starting to get them to move down these different pathways, okay? And so that's the bottom line. It's just a classic example because it shows us and names three very, very famous signaling factors that are teaming up. And developmental biologists love this because how do we get our complicated bodies out of the steel? And the answer is, well, a lot of it is the patterning that is regulated by cell communication. And this just shows that. And so we're just going to look at this and see this. So this figure's in your book, and you don't need to see it in detail, but just notice this, right? That the names of the morphogens are coming from this first source, the notochord, and what's called the floor plate of the neural tube. Part of the neural tube is sonic hedgehog, and I always just breathe it like that. So sonic hedgehog. Hey, isn't that important for your fingers and toes? Yes. This is just a different setting, and it's like it's a screwdriver, and it does that job there and this job there. Use it over and over again. So that's that one, a really famous one. And then coming from the upper part of the neural tube are winds. Like that. So wind, that we learned about, it can sometimes function as a morphogen. We just learned about it as a straight out, there it goes, it's gonna respond. But target cells can respond differently to different amounts of wind. And then the third one is BMP4. And that's a member, and this was a 1-point question on your exam, that's a member of a big family called TGF beta. So a big family of paralogs, like that. And these are so useful in development that in complicated bodies like humans, each of those is a gene family. So not just, different kinds of screwdrivers like that, but, in detail, different. So I think there are like 40 members of this TGF beta family in us. And again, it's just comparing like, at home, you just have a little set of tools because you're not an expert, but there's another person driving around with a pickup that's just got their whole back of their truck as a toolkit, right? So our body's like that, And so this is a specific family member. Sonic Hedgehog is a specific member. There are only like 3 or 4 of those. There are like 16 different winds, yeah?
So these three morphogens are all coming from the notochord, or they're all?
No, they're all coming from these physically different sources, like that, yep. And so we've got Sonic Hedgehog coming from here, and there's high concentration near it and lower as you go away. Then we have the winds, I said, I guess, went from here, coming from that region in the upper part of the neural tube. It's called the roof plate. And then we have these BMPs coming from the lateral mesoderm. And so we learned, the notochord neural tube, they do one thing. The somites do another thing. Lateral plate does another thing. But now we're just starting to introduce that as the cells are precursors, they're talking to other cells. Part of being a precursor of the notochord is inducing the neural tube. Part of it is also to help pattern the somites. So that's just a new idea, that these precursor cells are talking to each other, and that's part of, it's just like we go back to that fate, like where you're talking to your friends about, your fate, and you're hearing from them, they're helping you, find your fate, you're talking to them and helping them find their fate. And that's part of it. The early development has cells talking to each other, and it's back and forth, and it's ongoing. And the timing is critical, right? We're gonna do this, I'm gonna release some Sonic Hedgehog right now, that's gonna help you out. So we're helping those other cells out as they start to become different and go toward these, very detailed fates, right? Of all the different things the somite has to make, okay? So again, it goes back to chapter 1 where it says, here's a morphogen. And we have our good example of morphogen there. And then this is just an example of, whoa, morphogens can really do a lot, especially in a complicated organism like a human embryo and vertebrate embryos, because this is a vertebrate embryo story, okay? So questions about that, because that's this one kind of package. Just a little package of extra information focused on morphogen. Morphogens are only signaling factors, and only some signaling factors, and only some of the time. And that means that the target cells make different responses depending on the concentration they see. That's it. And we have those 2 examples. And we'll learn some more examples as we roll along. Okay, and we're doing great. I thought we'd get to some plants today, but probably won't, but maybe we'll get the rest of this covered. So here's another one that comes up in chapter 4, and we just want to cover it. And it's a certain group of genes that are called collectively homeotic genes. And there's a definition of this here, but we'll set it up like this. So, usually when there's a mutation, well, of course, it's always random. And usually a random mutation is going to create a hypomorphic allele, some loss of function, some decrease of function. So most mutations cause a loss of a structure, where it's not complete. They're missing pigment, an albino person, they're missing pigments. They lost the pigment enzyme. loss of function, okay? So there's some mutations that cause a really different phenotype. And instead of losing something, you replace it with the wrong thing. And that's what homeotic genes are. Homeotic genes are genes, when you mutate them, you don't lose something. You really aren't losing something, you're replacing it. So before we talk about definition, I'm just going to give you an example. So here's an example. This is a normal fly, and if you're looking at its head, it's got eyes, and it's got these little antennae, these little feelers. They're supposed to have antennae on their head, they're sensory organs. And this is a mutation in a homeotic gene, and instead of having antennae, they didn't lose the antennae, they got them replaced. And they're replaced with legs. Like that. So that's a homeotic mutation. And there are some genes that happens. When they get mutated, it's not like you lose the feelers, the antennae, you replace them. There are other kind of genes that are not homeotic where they could get mutated and whoops, no feelers, no antennae. You just lost them. None, didn't get them. Like that, they're defective. This is not a loss, it's a replacement. And that's a homeotic gene, and that's true for just some genes. And it's a bunch of genes, but just some of them. And so a homeotic gene is a gene that can cause a phenotype that's called a homeotic transformation. And that phenotype is caused when the homeotic gene gets mutated. When you lose its function, you don't lose the feelers, you replace them with the wrong body part, like that. So, and that's just one example. There are a bunch of different kinds of homeotic genes that can cause that, but they're odd. And the first person to study them was this guy named Ed Lewis in California and Caltech in Los Angeles in the 1940s. And it was a long, long time ago, and he was just Ed was just curious. Like, that's weird. This gene, when we read chapter 2, we'll learn the name of it, so we might as well mention it now. It's called antennapedia, and that's a pretty good name because its antennae are replaced by legs. Pedia can be like feet, legs, like that. So Ed is naming these genes, and he calls that antennapedia, which is a homeotic transformation from antennae to feet. And the next thing you say is, that's weird, you never see a human with this kind of mutation. And the reason is two that we'll learn about later in our course. One is we have duplicated those homeotic genes. So even if one is mutated, it's just not likely to cause a big visible problem. That's happens in us. The other is that if there is a homeotic mutation, a homeotic transformation in a human embryo, it almost certainly is going to die and not be born. So we don't see kids being born, oh, you got your legs coming out of your head, that's going to be good for sports, kind of, maybe soccer, because you don't do headers, just kick it, just keep kicking it, right? Right, we just don't see that because that would be such a dramatic birth defect that the baby is lost, as an embryo or a fetus early, a spontaneous abortion like that. Yep. Okay. But scientists can create those by knocking out multiple homeotic genes, and so we can see mice with homeotic transformations. An example of that would be a mouse that has ribs in its neck. So wrong place. You didn't lose anything, you're just making the wrong thing at the wrong place. And so that happens. And so these homeotic mutations are something very, very curious, but also significant. And so again, they were studied a long time ago, and just doing genetics, just kind of figure it out genetically, not understanding it, not being able to get kind of a grip on it. And this is a good example of how in the 1980s, when geneticists and biochemists started being able to manipulate these genes, they found out, oh, that gene is doing that function, and there are orthologs of antennapedia in every animal that has an anterior-posterior axis. There's a set of homeotic genes that set up the head-to-tail axis in every animal on our planet that has an anterior posterior axis. So those somites, they go 1, 2, 3, 4, 5. There's a set of genes that are setting that up. Those are addresses. And you can think of the legs growing out of the head, you're just at the wrong address. You're putting legs at the wrong address. They're putting them up here at the anterior end. They should be here. You know, not at the anterior end or the posterior end, but here. And that's not all homeotic genes, but there's a set of famous homeotic genes, and they're called Hox genes. So the Hox genes are a family of paralogs. Here they are in fruit flies, here they are in mammals, and they set up the anterior posterior axis. So we got to just talk about them for another 5 minutes on Thursday, and then we'll roll on. So now we got to get the exam back, and remember that